Good. All right. So calcium is the second messenger. All right. That's basically what we're doing. Remember, this is in the context of the master signaling mechanism was G proteins. We're going to see calcium in a lot of contexts. And as I'm saying, this is definitely the most intuitive. Unfortunately, I revised the lecture this morning and I didn't copy it onto my USB drive. So this is a couple days old version here. This is going to be our outline. We're going to look at calcium as an ion itself. And energy. I've been trying to give you guys a sense of energetics as we go. We're then going to look at calcium physiology, meaning what it does, calcium fluxes, how it increases and decreases in the intracellular space, and then the coupling of these fluxes to the physiology, and finally some long-term issues from these, some long-term events, and we'll. Okay. Here's from the periodic table. This is interesting. To me, this is interesting. So here are salts. So these are the cations. Here are the anions. And what you see is the ion when it's in its full electron configuration, and the red ones when it loses electrons, and the blue one when it gains electrons. All right. We're focusing here on calcium, obviously. And so the atomic size becomes much smaller when it loses its electrons. So because the electrons are gone, and so they don't occupy that outer shell. And so you have and of course the nucleus hasn't shrunk. The nucleus still has all of its protons. And so the electron cloud that is left is pulled much more tightly into this excess of two electrical charges, okay? And so the ion becomes smaller. Notice just by your eye, okay, that calcium is roughly the same size as sodium as when it's in the ionic state. So the calcium, although it begins much larger, and you can go by the numbers then if you want, although it begins much larger when it has all of its electrons present, it actually comes down to, if anything, even smaller than sodium, even though sodium begins smaller and it has lost only one electron. So the charge density around the nucleus is not as high. Now and look at how big potassium is, because it has all these extra shells of electrons, and you lose an electron, it's like, so what? It was loosely bound anyway, so it doesn't change very much, and then lithium is much smaller than everybody. Magnesium is a very small ion, okay? It has a very high charge density, and without any protecting layers, without any shells to mask that charge. So this thing comes down very small. The reason that I think these things are important is two reasons. First of all, not infrequently, the calcium and the sodium are going to move together. You have lots of ligand-gated channels which allow both calcium and magnesium in, and the answer is, well, then those must be size discriminating, because if they were charge density discriminating, it wouldn't let both in. It would see the calcium as just as a intense electron drawing, intensely positive charge. Okay, all right. And then the second thing is, just as I said, how I want you to think about the charge density that is in here as opposed to in here for the sodium, even though they're the same size. This is much more because you have because you have two positives. The consequences of that are all over the place, all right? So, for instance, you don't hydrate sodium that much. You do a little bit. But look at this. This thing is complex to 8 water molecules. And here I've put a diagram of water over here for you because it's in a tetrahedron. You have 2 lobes of free electrons, so highly negative. And as soon as you put them in the vicinity of this strongly positive ion, cation, they just orient themselves toward it. And they together then will go a long way towards shielding that charge. Okay? So these are all electrostatic interactions, and they're important. And they're important because now you plunk the calcium not in just water, okay, but now you plunk it into the region of the membrane or into the region of other proteins, okay? And look at, it's no accident that the amino acids that are here are things like aspartate, okay, which is going to be carrying a net negative charge because you have two carboxyl groups. So this thing has a net negative charge. So the water was simply a van der Waals interaction with the electrons orienting toward the calcium. But this is a real electrostatic interaction because you've got a negative charge and a positive charge. And so this is going to be held very firmly in place. And the consequence of that is that when you bind calcium, look at this. This is the same protein, all the same protein. So without apo and with calcium bound. Look at the changes in configuration. I mean, I don't want you to get into the specifics of this. I don't care that it's apo 100, which is a brain relevant protein, but it doesn't matter. Look at the change of the configuration. Look what happened to this green loop look at what it is in the presence of calcium. So this is a very strong cation. Okay, and when you bind it to proteins, it's going to exert substantial changes in the conformation of the protein. Okay, big changes that therefore are going to either activate or deactivate the enzymatic functions or structural functions of the protein. Okay, so that's the message that I want to get across here. Oh, this is current. All right, let's look at the physiology. Now, two or three times over the last 2 or 3 weeks, I have talked about the fact that the resting calcium concentration in the intracellular space of a neuron or astrocyte is into the 10 to the -7th range. That's a 10th of a micromolar. Okay? At peak, after cell excitation, you reach millimole concentration. That is a 10,000-fold change in the concentration of calcium. Okay, so signal to noise. Your noise level is in the sub-micromolar range, and the signal is in the millimolar range. 10,000 times. The signal is 10,000 times the resting noise. Okay, this is massive, and a very strong cation that can exert huge changes in protein. This is so much calcium coming into the cell that you actually change the pH of the cell, because the calcium is going to displace protons off of the proteins, which is going to then make the cell acid. So these are huge changes when the calcium comes in. And so then we're going to use this for things like excitation, contraction coupling, and signaling of various ways, and long-term cellular changes. And that's what we want to look at in this talk. At the heart of this whole thing in the central nervous system is the voltage-gated calcium channel, not just in the central nervous system, all over the place. So we're going to spend some time on this, a fair introduction to this creature. They're nearly everywhere. Wherever you have excitable cells, you're going to find voltage-gated calcium channels. They are, in many ways, the hallmark of the excitable cell. Their structure is going to vary according to what they do. And the structural changes will allow basically the resting membrane voltage at which the channel opens, and also the amount of calcium that enters once the channel opens. So that's what you're going to do with the regulatory proteins or peptides that are associated with the voltage-gated calcium channel. You're going to be able to modulate, if you've got it, the central nervous system modulates everything, right? So all of the influences that we have been talking about through G-protein coupling, and that we're going to talk about next week also, they all are going to have the ability to modulate the voltage-gated calcium channel, all right? You're probably familiar with the letter nomenclature, L-type, T-type, N-type, Q-type, that sort of thing. But this was the way it was originally set up, and we don't do that anymore. That's turned out not to be the best way. We'll come to that. This is just a schematic view of the voltage-gated calcium channel. Your alpha-1 subunit is the pore that allows the calcium in. And then we'll look at some of this. We'll have a beta regulatory, which is intracellular, a gamma, which is in the membrane, and alpha-2 and delta, which sit actually on the extracellular surface of the membrane. This also anticipates where we're going with this as the calcium comes in. This is obviously schematic. It's not a single cell. You could use it for actomyosin activation if you're in the muscle. You could use it for secretion coupling if you're in a synapse, in a presynaptic terminal. You can, anywhere that you are, you can use it for protein phosphorylation, and you can go ahead and change the nuclear structure if you want, depending on the levels of calcium, and that's, we'll talk about that. So this is the way things would have looked 15 years ago, maybe, all right? And here are the organs where they're first found. And notice that the voltage of activation, the reason that I keep this slide here is it's good. It shows you that they have different voltage activations and different conductances. So an L-type channel has a very high voltage activation. You don't open this channel until basically the action potential has already started in a neuron, okay? And that's just the way it is. So this is minus 10, right? Minus 70 is the normal resting membrane potential in the nervous system. So the T-type channel is active down here at that resting membrane potential, right at baseline. And it only lets in a small conductance. So this is a nice way to keep the cell paying attention to the world, right? Very small sips, always sipping, always trying to sense the environment, okay? That's here. This guy, you know, explosion, open the floodgates, let the thing in, and then get rid of it, okay? Boom. This is an all or none type of channel. This is a very sophisticated and subtle little guy, and the N type and all these other Q types are in between, okay? Again, just the subunits that we talked about, alpha 1 is the pore, and notice. Now you say, well, okay, you think back to the red piece of the ionic, periodic table that I showed you. Not surprisingly, the same alpha 1 core that lets the calcium in also is what's used for sodium channels. So evolutionary, back to evolution, okay? It makes the right size pore. And the calcium ion and the sodium ion are the same size. Okay, so if you don't do something else to discriminate, meaning sense the charge density of the cation, then they're both coming in together. Or the same pore that you use for one can be used for the other. Okay, so they're very similar, evolutionary. But they're not normal. That makes the channel, but you can't do anything with that channel. You wreak havoc with the cell. And so now what you do is depending on what cell you're in, depending on the function of the cell and the recent history, you use these other channels. The beta subunit gives it the voltage sensitivity that you want it to have in that particular cell, and also serves for gating purposes and that sort of thing. The gamma subunit, which was in the membrane, is related to trafficking within the membrane. Again, think back 10 lectures now in this course. We're very far along, and you're creating these big membrane domains, or I use the term rafts over and over, where you assemble protein assemblages to have an ongoing active function where they all interact with one another. So you have to be able to move this subunit into those kinds of functional units, which you do with the gamma subunit. And I don't care that you remember these names. The point is it has all the functions that it ought to. You have others that regulate voltage sensitivity and others that anchor it in place. Okay, it's like this is the basic kind of stuff that we're doing for the last 10 weeks, but now we're looking at it as a voltage-gated calcium channel. Here's another view of it, showing you that it has these intramembrane loops and transmembrane loops and all the things that we have previously discussed. And here are the peptides that are available to modulate it. That's what's important. What's also important then is that the different alpha 1 subunits divide voltage-gated calcium channels into 3 different families. And this is where you go back to the L type, the T type, and all the others, NP, Q, R, X sort of type, okay? But now we give them new names, okay? So CAV-V means voltage-gated calcium channel, type 1. That is equivalent to the L type. CAV3 is the T type. CAV2 are all of the NPQR types. Okay? We say, well, why did we do that? Okay, because there's more to the story. Okay? Because now we can begin to differentiate the families of CAV1, CAV2, and CAV3 with a meaningful nomenclature here, like this, with the decimal system. And notice that all of the CAV1s are similar in that they are not central nervous system. They all are explosive types of kill the resting membrane potential, flood the gates, and very slowly deactivate the system. That wouldn't work well in the central nervous system. Type 3 are consistently pacemaker. The T-type channel is an oscillatory channel. Remember, that was the very low voltage, very low SIPs. This thing is perfect for setting the cell ready, not ready, ready, not ready, oscillating, pacemaker function, suprachiasmatic nucleus, atrial node, okay, T-type, and the relay neurons, most importantly for us, the relay neurons of the thalamus, okay? Cav2 family are the central nervous system. All the things that go on here in various different cell types are different variants of the Cav2 family. Okay? All of this is voltage-gated calcium channel. All of this, okay? All right. So let's see it in action, all right? Let's get to cellular fluxes, all right? We start with an extremely low calcium level. I keep emphasizing extremely low. You would have to work incredibly hard to clean up. If I want salt, when you have salt at the table, believe me, you have more calcium in your salt than these cells have in their cytoplasm. Okay, it's hard to separate calcium from sodium because they're the same size. Okay, so your table salt has lots of calcium in it. Okay. Anyway, then what's going to happen, and here now we're looking to, so we're talking about flux. We're going to start with influx. How does calcium get from outside the cell to inside the cell? So you have two possibilities. One is the voltage-gated calcium channel, and the other one is ligand-gated channels. Ligand-gated channels, all right? So that's what we're going to be looking at. You have either ligand or voltage-gated calcium channels that open and permit some calcium to come into the neuron or the excitable cell. And the calcium actually crosses the plasma membrane and enters the cell. Okay? You can do that. Here are some of your ionotropic receptors, the glutamate receptors, acetylcholine, GABA. All of these, including ATP here in your P2X sets, let calcium into the system. They're ligand-regulated calcium influxes. This is a voltage-gated. We've been talking about the voltage-gated calcium influx. So you can change the voltage of the resting membrane potential and open up channels depending on what the channel is and how it's set up. Or you can bind the ligand to a receptor and you can open the calcium gates and let the calcium in by a ligand control. Once you do that, no matter which way you let it in, once you do that, you're now increasing the calcium, and that will set in motion an amplification process. Okay? It may, first of all, open more voltage-gated calcium channels and let more calcium in, but it also may release calcium from intracellular stores. There's huge amounts of calcium that are stored in the endoplasmic reticulum or in the sarcoplasmic reticulum. Okay? So those are filled with calcium. All right, and then let's go back just one lecture, and let's talk about phospholipase C. What did phospholipase C do? It cleaved the phosphoinositol into the I-145 and the DAG, right? The DAG activated protein kinase C, but what did the IP3 do? Open calcium channels, right? So anything, all those metabotropic receptors that we saw, that activated phospholipase C are going to set this system up for an amplification. And I hope you also remember that most of them required a little bit extra calcium along with the phospholipase C. Here you go. Here it is, okay? In you go for letting to open this, to start a little calcium, prime the pump with a little bit of calcium. At the same time, you're probably generating IP3. IP3 is going to wander over to the endoplasmic reticulum, and in the presence of a little bit of extra calcium, open the system and let the calcium out of the intracellular storage sites. So that's what's happening. That's the signal amplification. That, remember, we've got a 10,000-fold amplification of our calcium here. All right, so a little bit of calcium in, and then that goes on. Then you release that via the metabotropic receptors, and this is what we just said. This is it, okay? So we don't need to talk about it anymore. This is a nice diagram. We're going to use this diagram several times. We're only interested in the rising portion of the calcium curve right now. So here you are. I like this because you can have a receptor-activated calcium channel. You can have a voltage-gated calcium channel, or you can have a G protein coupled receptor, which turns on the phospholipase C, generating the IP3, and in the presence of calcium, you then open what is called the ryanodone receptor, which is in the endoplasmic sarcoplasmic reticulum. And when you do that, this ryanodone receptor, you just release massive amounts of calcium into the cell. So maybe this stuff will raise the calcium level from 10 to the minus 7th to 10 to the minus 6th. And then this does the job from 10 to the minus 6 to 10 to the minus 3. Okay? And that's where it's going. And we'll get to all the other parts of it, getting rid of the signal and buffering the signal and everything. We'll get to deal with all of that. You end the release, okay, when the levels in the cell hit about 10 to the minus 5th. So they're going to keep on growing, but you're going to start to close the system around them. These channels, both the IP3 and the straight ryanodone receptors, are going to start closing. You do buffer. The cell has lots of ways of buffering calcium. One is the mitochondria. The mitochondria has what's known as the uniporter. It can take up, when the calcium levels get high, the mitochondria soaks it up. Okay, it doesn't just enter non-specifically, it comes in through this uniporter. You also have in the average neuron over 200 calcium binding proteins. Now, those, I wouldn't want you to think of a calcium binding protein as a calcium sponge. Clean up this mess, okay? They're all activated by the calcium. There are just so many proteins that are activated by the calcium influx that it actually quantitatively serves as a buffer for the system. But that's not what they're there for. They're not a buffering system. Okay, they serve as a buffer, but that's not what they're for. What they're for, okay, is to be activated by the influx of calcium. But this tells you how many proteins are going. So think about it, the average central nervous system neuron, it's firing at a minimum 8 to 12 times a second, okay? And so this is going on all the time. It's being activated through presynaptic terminals, and you keep letting a little bit of calcium in. The T channels are going, yeah, a little bit. Then at some point you get to the point where when it's when higher concentrations than 10 to the minus 6, you start activating all of your other channels, and you open the ryanodone receptors, and suddenly you have an action potential. Okay? So that's the way this is going. Here's your uniporter, just cartoon diagrams. Here's a regulated, all of it modulated, all of it. By regulated, I mean at the moment. By modulated, I mean, over a matter of minutes. In it goes, calcium going into the mitochondria. And I also have this here because gap junctions allow calcium to be shared among the astrocytes in particular. And so if one cell has a high concentration, it can just let some of the calcium into another cell. The astrocytes function almost like a silicium at times. Then you have to get rid of it, okay? If you're going to have a signal, you can't only turn it on, you have to turn it on and you have to turn it off, okay? And you turn it off by lowering the calcium concentration again. I used an analogy before when I was talking about vacuum pumps in the lab when I was working in the lab as a graduate student, and saying that when your system is flooded with 10 to the minus 3 calcium, okay, you can't use the same pump to get you from 10 to the minus 6 to 10 to the minus 7th as you would use to get from 10 to the minus 3 to 10 to the minus 6. 10 to the minus 3 to 10 to the minus 6 has to have two properties. It has to have a huge capacity, right? Because there's a massive amount, okay? And it can't be expensive energetically. That's the second property. I mean, if you had to pull every single calcium molecule out, okay, that would be very expensive action potential. You'd burn the system up in no time. So you have to have a very efficient way But that efficient way, which doesn't expend a lot of energy, is not going to get you down to the lowest levels. Okay? In order to get down to the lowest levels, you're going to have to expend some energy. But here, at least, you don't have a lot of work to do. That is, you don't have that many more atoms that have to be removed. And so you can use an ATPase to get you down to 10 to the minus 7. So you integrate the system. You have, in order to, the high capacity system is a sodium calcium exchanger. You exchange calcium for sodium, and that's all you do. And you get rid of your calcium that way, and that's called the sodium calcium exchange protein. And then to get it down to the lowest levels, you use a plasma membrane calcium ATPase. And you expend your ATP, and you get the calcium back down to the low level that you need. And you do a similar sort of thing with your reuptake system. The uniporter of the mitochondria sucked up a lot of calcium very easily. But in order to get everything back into the endoplasmic reticulum, you're going to need a sarcoplasm endoplasmic reticulum calcium ATPase, circa. Okay? And you integrate the whole system, obviously. Okay? But there's one more problem, okay, in ending this signal. If I'm extruding, right, so most of the calcium that is flooding the cytoplasm is coming out of the endoplasmic reticulum, right? So some of it's coming from extracellular, but not that much. Most of it is from intracellular stores. But then we're removing the calcium, we're getting down to a low level with this sodium calcium exchanger, which is causing the calcium to leave the cell. So what's the problem? If you do this repeatedly, what's the problem? Where you don't have enough calcium in your endoplasmic reticulum, you're going to deplete the endoplasmic reticulum. You let it out of the endoplasmic reticulum, you throw it out of the cell. Pretty soon, even though you're taking the calcium back up, some of it is being lost to the cell. And so these are known as the stores, your calcium stores. So you have a store-operated calcium channel to replenish the system. Look at this. So this is a great diagram. I've added a couple. High affinity, low, high affinity, but low transport. I don't know what I was thinking. Low affinity, high transport. Right, so I've put those in for you. Same thing here, high affinity, low transport. But then here you have the circa, should have been low affinity, sort of high affinity and low transport. That's correct. So it's pulling that in. But you're going to lose it. And so I like this diagram. I like this diagram because it begins to enter same characters, but in this case, you're introducing the modulatory systems, the cyclic AMP modulation of the ryanodone channel, of the circuit channel, all of them being phosphorylated, dephosphorylated. You can regulate all of these things, okay? And then this is the stores. So look at the beauty of this. Here's your endoplasmic reticulum. This is what it normally is. The endoplasmic reticulum is not near the plasma membrane. It starts to run out of calcium. It needs to send a straw through to the extracellular space and suck the calcium back in. And it literally does that. It literally takes a channel, which is normally not connected to anything, moves its endoplasmic reticulum bilayer to the plasma bilayer, okay? And then actively connects this store protein to a calcium channel and just lets the calcium back in, refills itself. I mean, is that not cool? It's very cool. I mean, this is an amazing system. Okay? Now, go on to excitation, contraction, coupling. So we're getting these floods of calcium, and the question is, what are we doing with the floods of calcium? And from our point of view, how are we doing it? Okay? So the Cav1 channel initiates excitation, contraction, coupling. We said that. It's outside the central nervous system, and it's not the T cell, T-type. Then it's going to be muscle, heart, those sorts of things, chromatin tissue, that sort of thing. We'll all use this. The interesting thing here in skeletal muscle is that you don't actually have calcium enter the cell. to activate the release of calcium from the sarcoplasm. That's peculiar. So in the neuron, you have to raise the calcium level in the cell in order to release the calcium. But in the muscle, you don't. In the muscle, the change in resting membrane potential all by itself is going to result in a release of calcium from the sarcoplasmic reticulum. Okay? These Ca1.1 channels are in the transverse tubules, and they are directly sensitive to the resting membrane potential. So here you go. Great picture. End plate, acetylcholine receptor, resting membrane potential changes, travels into the T tubule, okay, directly influences the sarcoplasmic reticulum to release its calcium. Great picture. And then once it's loose, actin-myosin activation. Okay, great picture. In the heart, that's not true. In the heart, in the T-tubule system, the calcium has to enter, and in order to cause the release of the calcium from the sarcoplasmic reticulum in heart. But in skeletal muscle, no calcium entry. All right. So those are the main differences. Different channel, okay, different channel. And in the sympathetics, the main thing about the sympathetic influence, of course, is just back to that diagram that we saw back here, this one. And what makes the heart, we start levofed, right? I mean, isn't that what we're doing? So, okay, we're starting levofed in order to stimulate this G protein to generate cyclic AMP, which will cause more release of calcium from the sarcoplasmic reticulum to strengthen the cardiac contractility. That's what we do. That's our management. Okay, so the catechol, we saw a guy today in clinic who had a dissection, an intracranial internal carotid artery dissection, because he was stressed out. Okay, so those things happen. All right. The Ca2 family then is in the central nervous system. That's going to be about synaptic function. All right. And so you got this family, which is the Ca2 family, and we're then going to divide this all up into different subgroups of the family, which in the end are going to result in not excitation contraction, but excitation secretion coupling. And that's what's going on here. And we're going to modify all of it by voltage-gated calcium channels, if you want, by metabotropic receptors, and all of this gets modified. And you're modifying the calcium release and the calcium uptake that generates different levels of calcium as the action potential comes down and causes a release of the neurotransmitter, or not. Right? And this is back to where we were, again, 9-10 weeks ago. And I like this picture because you also modify the post-synaptic cell, and the calcium we're going to see in a minute, going to activate kinases and phosphatases, and it's just the whole thing is regulated. And the cyclic nucleotides through the adrenergic receptors constantly influencing the release and uptake of calcium, which generates the force of release or the contraction. Finally, you have the T-type channels, which is the Ca3 family, and these look open at the same resting membrane potential as the sodium channel. They are very fast. They're very voltage dependent. They're ideally suited to doing things like oscillating, so rhythmic firing. SA node, relay neurons of the thalamus is where they hang out, highly regulated by catecholamines and other neurotransmitters through G protein mechanisms. So I hope by this time the whole system is coming together, and you're going, I get it, like move on. Okay, so we'll move on. I wanted you to have a sense of the relative size of charge that enters the cell, sodium versus calcium. So even though the calcium cation is a dication, divalent cation, and is intense charge concentration, not as much not as much calcium is entering the cell as is occurring with the sodium. All right, and so you can see here how much larger the sodium influx is than the calcium influx. And you also see that the sodium influx precedes the calcium influx. This is higher mag, higher resolution, if you will. This is okay. Here's a Cav3 knockout mouse. Okay, so this particular mouse does not have a Cav 3.1. All right, look at this. If you take the resting membrane potential to -60, we're presuming -70 to -80 resting membrane. Once you bring them up to -60 and the Cav3 is missing, you can't see any difference here, right? They look the same, right? That's because that's not where the Cav3 is active. The Cav3 is relevant at -70. So let's look at -70 resting membrane potential and see what's going on here. This is the knockout mouse. See what happens? In comes a depolarization, and in the wild type, you get spikes. But in the knockout mouse with the Cav3 channel out, even though you have the same level of depolarization, you don't get any spikes. Okay, same thing at -80. In fact, at -80, these levels of depolarization produce polyspikes, and you almost get nothing if the Cav3 channel is missing. Okay. You have postsynaptic, just to say what at this point is probably obvious, but let's be clear that you can also have postsynaptic ligand-gated calcium channels. That goes without saying. That's basically what your AMPA receptor is. Okay, you can control different cell types, have different alpha-2 subunits. Okay, different glutamate receptor subunits. Everything gets regulated according to the structure of the voltage-gated calcium channels, as well as their ability to be modified by G proteins and other signaling systems. Now, the NMDA channel this is a great time right here in this calcium discussion. It really is a great time to talk about the NMDA channel, which is really, really different than AMPA. The AMPA receptor channel is very sensitive to, it's now. You put some AMPA on, you put some glutamate on this channel, it opens now, it lets the calcium in now. You take the glutamate away, it closes, and the signal is over. That is not the way it is with the NMDA channel. NMDA channel is a very different beast, okay? Here you have the NMDA channel. It is a slowly inactivating channel. Its activation requires depolarization and binding of glutamate and binding of another amino acid neurotransmitter, either serine or glycine. You also have to get rid of the magnesium because at resting membrane potentials, magnesium inhibits. The magnesium was a smaller ion. It has a higher charge density than calcium. It is the natural antagonist of calcium in the central nervous system. And having done 15, 20 years of research trying to protect brain slices, hippocampal brain slices, from ischemic injury, I'll tell you that there's nothing that I have seen that is more potent than magnesium. Okay? We were stunned when we put magnesium in the bath at high concentrations. It's an extraordinary neuroprotectant. Okay, extraordinary. Huh? Yeah, it's really potent. Okay, and it is the natural antagonist of the calcium. All right. And the other thing about an NMDA channel is you don't usually activate it at the same levels of calcium influx. It takes much more high levels of calcium to open the NMDA channel than it does. And so this diagram sort of summarizes all of that, okay? At physiologic conditions, the glutamate release is going to activate an AMPA channel, but at physiologic conditions, it's not going to activate the NMDA channel because it's blocked by magnesium. Okay, you're going to have to get rid of the magnesium in order to open the NMDA channel, and you're only going to do that when you have sustained elevations of calcium. That's the point. Okay, that's what makes the NMDA channel Okay, so all of this is saying what I said, and we've been over all of this. Okay, so let's just keep going here. This is a very busy diagram, but I like it. I like it for its complexity. Look here. Here are all three glutamate channels adjacent to one another, and they're showing you the different relationships of these 3 glutamate channels. This is the AMPA, excitoamino acid terminal. Okay? So very good diagram. So let's finish up with the long-term events of calcium influx into the cell. It has been known forever that high levels of calcium will regulate gene function. That's long-term. Gene function is definitely long-term. Because of persistently increased calcium levels, you change the number of synapses, you change the structure of your dendritic tree. You get growth factor signaling comes in this way. Post-tetanic potentiation, long-term potentiation. All of these are going to be long-term sustained levels of calcium in the system. Memory consolidation. And these are all going to be mediated through the NMDA channel and the voltage-gated channel, calcium channel. When calcium comes into the cell then, okay, in addition to all the other things that we've just been talking about of excitation, contraction, excitation, secretion, okay? In addition, the calcium is going to be sensed by calmodulin. So now we're talking about long-term events. This is the 3rd major category of what's going on in response to calcium, okay? You have you have calmodulin binding proteins, calcium binding proteins is a calmodulin. But it's not called just calbindin, but calmodulin because this peptide is capable of modulating the activity of enzymes. Okay? So it's a calcium calmodulin system. That's abbreviated as this way, CAM. And the calcium calmodulin system stimulates a calcium calmodulin kinase. And this is its effector. And also the calcium calmodulin kinase, calcium calmodulin also controls the function of the RAS proteins, which were small g proteins. Small g proteins, right? So the system is just starting to come together here. And next week, we're going to study kinases and phosphatases, assuming that I can get my act together because it's a massive lecture and very complicated. We're then, in addition to all this, we're going to activate the threonine kinases. We'll talk about that hopefully next week. Phosphorylation of transcription factors, CREB, nuclear factor kappa B, all of these things. And not only that, this system is capable of activating histone acetyltransferase, which means it's going to open up the histones so that the DNA can be exposed and then transcribed. Okay, translated, I suppose. This is a diagram to show you that calcium and calmodulin come together to make calcium calmodulin, which activates the kinase. This then can control the circuit, okay, which was, remember, the endoplasmic reticulum calcium ATPase. Well, maybe it inhibits it. Maybe it makes it more active. If it makes it more active, then the signal is going to be shorter. If it makes it less active, then the signal is going to be longer. Okay? I mean, it's infinitely complicated and much more than I want to try to do. This is a nice diagram because it shows you here's an NMDA channel, there's voltage-gated calcium channels, and they come either way. You get a lot of calcium into the system. You do this one and you activate RAS genes. You do this one, you activate calcium kinase, okay? You come in and you modify the nucleus, the transcription of the cells, okay? With these calcium calmodulin kinase 4. Okay, as a activation of transcription factors. Long-term stuff. This is what I'm saying. By integrating all of this stuff, you now are capable of all of this sustained elevation of calcium, which in the end will even result in apoptosis. And the oscillations for pacemakers, that's your CAV3. So we've, the calcium oscillations are sort of interesting. And the main thing about an oscillation is, I mean, what do you do with an oscillation? It's amplitude and frequency, right? Any oscillator, amplitude, frequency, and phase. Phase is the 3rd parameter. What's its relationship to another oscillator? Okay. Is it in sync? Does it have the same frequency or not? Okay. Is it going to have a destructive or constructive interference? How big is its amplitude? All of that is going to be regulated through because the oscillators are going to be calcium oscillators. So your ability to regulate the influx, the release, the uptake, the extrusion is going to control your rate of oscillation, your amplitude of oscillation. If you do it very slowly, this is always true, okay? I turn the faucet on in the sink, and then I turn it off, and I wait for the water to drain out of the sink. And I turn the faucet on again, and I wait for it to drain. And that's what I get. If I turn the faucet off, I turn the faucet on, turn it off, but don't wait long enough, turn it on again for the same amount, turn it off, turn it on again, but don't wait long enough for it to drain, turn it on, pretty soon the sink's going to overflow, even though it's only oscillating. I'm not running it continuously, but I'm running it too fast for the system to return to baseline. Okay, in that case, this high frequency calcium oscillation is going to accumulate calcium. If I accumulate calcium in a neuron, then I end up with a post-tetanic, so the tetany is the high frequency stimulation. The post-tetanic facilitation or potentiation is because I let too much calcium into the system. See, so all of this physiology that you all studied last year, now you're looking at the mechanism by which this is occurring, okay? And so a high frequency action potential train is known as tetany. The tetanic stimulation results in elevations and saturation of the calcium stores and levels. And once you saturate the system, any more signal that lets in any more calcium can't be buffered. So the net effect of the same number of calcium atoms entering the cell is going to be much greater after a tetany, because all the buffering mechanisms are done. The system is saturated. Okay, so now you let some calcium in. It's raw. It's unbuffered, unextruded, raw in it goes. Okay, and that results in a much larger release of the neurotransmitter at the presynaptic terminal. And that provides you with short-term memory. That facilitates that neuron group. That neuron group, which was exposed to, it doesn't matter whether it's tetany. Tetany is in a lot of ways an aberrant activity. That's something that we do in the lab. The point is that if you strongly stimulate a neuronal assemblage, okay, the neurons in that group are going to have rises in their calcium level. Okay, and when that happens, they're going to be facilitated. And if you keep doing it, you're going to have a much stronger release of neurotransmitter, and you're going to facilitate the postsynaptic. Of course, you can get to the point where you totally saturate the signal and it won't turn off, at which point you lose signal, and that is fatigue. All right? So this is a great example. If you don't do a tetany, so here's your spiking, okay? This is your presynaptic spiking. So here's your tetany. Here's the postsynaptic response. So here's your tetany. And these guys get bigger during the tetany, and then they keep getting bigger, and then they come down. The time course, which doesn't matter what it is, it's much longer than the tetany itself, the single action potential, right? So this is post-tetanic potentiation. It's a calcium phenomenon. You're saturating your calcium stores, and that's all there is to it. And if you think of it that way, then it just makes so much sense, okay? And when you do that, that's when you open the NMDA channels. That's exactly that's the kind of signal that the NMDA channel is waiting for, not the little that the AMPA receptor is living with, but it's build up, build up, build up, build up. What's going on? All this calcium, sustained levels of calcium, get rid of the magnesium, open the NMDA channel, and now you can modify the cell. Now the cell is going, I didn't have the capacity. I'm being used more than I have the capacity. I need to make more of this, make more of that, grow more of this. This is fun. I'm being used a lot. Okay, I better modify my structure and be capable of handling a much higher volume of information trafficking. Okay, and so it needs to, in order to do that, it's going to have to transcribe genes. In order to do that, all this signal has to be, and that's what we were talking about before. All of this is done through things like calcium, NMDA channels, calcium calmodulin kinase, activating gene transcription, and you're on your way toward growing a bigger neuron, more dendrites, stronger dendritic tree, smarter child. Stimulate the child, okay? Enrich his environment and make him smarter, okay? Deprive him and he ends up dull, right? I mean, that's what we're talking about at the level of the calcium channel. Give it too much, like we do in ischemia, give it much too much and sustain it for too long, and you set off the apoptotic pathway, which is also calcium signal pathway. I don't want to get into apoptosis. And one of the few real obvious and direct clinical syndromes that we have that are related to an abnormality of calcium is the Ca3 abnormality, 3.3 in the thalamus, okay, which results in classical absence seizures. Okay, it's an inborn error of metabolism involving the Cav 3.3. Okay. And that's what's going on in there. And it's an oscillatory problem, right? So it's an oscillatory channel, a Cap3 family channel. Job is oscillation. You end up with an oscillatory abnormality because the channel is miscreated. Okay? So that's calcium. That's calcium, folks.